Assalamualaikum warahmatullahi wabarakatuh and good morning to everyone and welcome to our research proposal presentation for SSCT 2423 Research Methodology in Biosciences. My name is Wan Adrina binti Wan Ahmad Azmi from Group 1 and I will be presenting our study which is titled Bioremediation of Potential Gram-Negative Bacteria Against Oil Contamination in Tanjung Langsat, Pasir Gudang. This research explores the potential of soil bacteria to naturally clean up petroleum pollutants in critical industrial areas of Johor, Malaysia. Next, these are our contents that we will be discussing for our presentation today. Background study, problem statement, objectives, flowchart of research, methodology, Gantt chart, significance of study, expected outcomes, and references. Industrialization brings progress, but also pollution. One of the most pressing environmental threats is oil contamination from petroleum-based industries. In Tanjung Langsat, oil spills have already been reported, threatening both soil quality and marine ecosystems. Instead of relying on expensive or harsh chemical cleanups, bioremediation may offer a biological solution, which uses microbes such as bacteria to break down harmful hydrocarbons into harmless substances like carbon dioxide and water. Among the most promising are gram-negative bacteria such as Pseudomonas acinetobacter and Enterobacter, all of which are naturally found in soil. Although many microbes have the ability to degrade oil, not all of them are equally effective, and unfortunately, there's not enough local data on how different bacteria strains may perform in complex soil environments like the ones in Tanjung Langsat. We also don't know yet which bioremediation technique, which is bioaugmentation or biostimulation, will work best in this context. Our research aims to bridge this gap by identifying suitable bacteria, optimizing treatment methods, and analyzing the effectiveness in degrading petroleum contaminants. Therefore, we have broken down our research into two main objectives. First, to assess the level of oil contamination in soil from Tanjung Langsat. Second, to evaluate the oil degrading ability of gram-negative soil bacteria using bioremediation techniques. Here is a simplified version of our research workflow. First, we will begin soil sampling and oil contamination analysis using GCMS. Next, we will isolate and identify gram-negative bacteria through biochemical and molecular tests. After that, we will apply two bioremediation techniques: bioaugmentation, where we will add selected bacteria into contaminated soil; biostimulation, where we will boost the activity of native bacteria by adding nutrients. And over 30 days, we will monitor the changes in total petroleum hydrocarbons or TPH to measure how effective each treatment method is. Our soil sampling will involve collecting 20 samples from Tanjung Langsat and storing them for proper analysis. We will use gas chromatography mass spectrometry or GCMS to detect and quantify hydrocarbon levels. Next, we will isolate and identify 3 gram-negative bacteria: Pseudomonas acinetobacter and Enterobacter. For bioaugmentation, we will introduce these bacteria into separate soil treatments. In biostimulation, we will add nitrogen and phosphorus to encourage the activity of indigenous bacteria. Sampling is done at regular intervals on day 0, day 7, day 14, 21, and 30, followed by GCMS analysis. Finally, we will apply statistical analysis like ANOVA to compare the degradation performance across all treatments. Next is our Gantt chart, which will center around our research schedule to run over 10 months. In months 1 and 2, we will focus on planning and literature review. Sampling GCMS analysis take place in months 3 and 4. Isolation and identification of bacteria are done during the months 5 and 6. Bioremediation experiments are followed by in months 7 and 8. These final 2 months are dedicated to data analysis and proposal writing. This timeline will ensure enough time for lab work, analysis, and proper documentation. Next is the significance of study. This study matters because oil spills don't just damage the environment, they will also affect communities and livelihoods. By understanding how local bacteria can be used to naturally clean up pollutants, we will provide a sustainable and low-cost solution to a serious issue. Our research could not only help Pasir Gudang, but also other industrial zones facing similar challenges. Next is our expected outcomes. By the end of our study, we aim to identify which bacteria species is most effective at degrading petroleum-based pollutants, determine which bioremediation technique, either bioaugmentation or biostimulation, offers the best results under local conditions, and recommend a practical and scalable strategy for future environmental cleanups. Our proposal is backed by recent studies and journals, including these works. A full list of our references is included in our written proposal. Thank you so much for taking the time to watch and listen to our video presentation. We truly appreciate the opportunity to share our research and environmental concerns, and we hope to address this through our study. Bioremediation may seem like a small step, but in the bigger picture, it's a meaningful approach towards healing environment, especially in places like Tanjung Langsat where nature and industry are closely coexist. We also hope that our work can inspire future research and spark more sustainable practices in dealing with pollution. Once again, thank you for your attention, and if you have any questions, we'll be happy to hear them. Have a great day and take care, and Assalamualaikum warahmatullahi wabarakatuh.